So good afternoon to everyone. My name is Vinit Yadav from Electrical Engineering Department in IIT Delhi. And here my presentation topic is in vitro localized entrapment and targeted delivery of magnetically loaded anti-cancerous drug on magnetic lab on chip. So this is the outline which I'm going to discuss in my slides. We'll start from the introduction, then motivation, development of a magnetically controlled drug, then device design and operation, then compartmentalization of the drug and experimental results, and the last, the summary. So as we know that nowadays cancer is a fatal disease which is rapidly growing nowadays widely, and also it's causing death naturally. And due to that, what is the cancer? Cancer is like abnormal cells which is uncontrollably it is invading to very healthy tissues. So there are some traditional treatment methods by, like, for example, surgery, therapy, radiotherapy, and chemotherapy. This is the traditional method by which we are doctors are treating the cancer patients. But there are some limitations of this conventional drug delivery systems because it contains the low half-life, high volume distribution. If you talk about the pharmacokinetics, and for example, if you talk about the pharmacodynamics, then low specificity and low therapeutic index. So, and if you talk about the therapeuticity, then the high dose and adverse effect has been also tackled in such conditions. So there are some solutions by the researchers for these particular targeted drug delivery. That is, a lot of kinds are there, but here I am talking about two kinds. First one is the active targeting, and second one is the passive targeting. Definitely in passive targeting, we are not using any source, and in which the EPR effect is going to work. But in active targeting, we are particularly using some kind of source like some electric field or some magnetic field or some kind of thing. So, but before the treatment of the device cancer, if we have some device by which we can find out which drug is very important for that particular person, then definitely it will boost the treatment methods. For example, if any patient having some bacterial infection, so in general, doctor prescribes 3 to 4 type of antibiotics, but why not one? So if we have a device in which we can culture some cancer tissue of the patient, and if we inject a drug, then which drug is more effective for that particular patient? If we can find out, then definitely it will be more beneficial for our society. So in this regards, here there are some manual trials and testing methods are also available by many researchers. But in manual trials and testing methods, what is the limitation? There is some manual error, and also the consumption high, and also the reagents are also very highly used. But if we talk about some lab on a chip application, and particularly if you focus about the magnetic lab on a chip, because in magnetic lab on a chip, if for depending on the permanent magnet, it don't take any electrical circuitry, and also it can like portable kind of thing. So we can take from in one lab to another lab. That's why here I am talking about only magnetic lab on chip. Definitely, most of the other sources are also very important, but here specifically, since my research work is based on the magnetic lab on chip, that's why I am just focusing on this. So most of the magnetic lab on chip is depending on to the separating the WBC, RBC, or some magnetic nanoparticles, or some magnetic biodetection or drug diagnosis, because it's precisely control over the fluidic environment, enables the use of fewer reagents, and also it reduces the cost and time, and various functions can be done on a single chip. In magnetic lab on chip or in cancer treatment, magnetic nanoparticles have been widely used. What they're doing, the researchers have doing they are tagging some kind of drugs with the magnetic nanoparticles, and they are magnetically controlled or magnetically tuned at a particular location to the drug without any external circuitry. So by these motivations, my goal is that to prepare some magnetically controlled drug, and also for testing that drug to prepare some magnetic microfluidic device, and also in order to find out which drug is important for the person. This is also highly important, that what should be the correct dose required for the person, because as we know that according to immune system, for the same disease, might be one patient required for a lesser dose, some patient might be required for a higher dose. It's totally depend on the immune system. So if you have also device by which we can find out, okay, which concentration or which dose is fine for that particular person, then it will be also good. So it's also part of my research work. And so the device should be also susceptible to culture different cells. It can be like cancer cell, or it can be like normal cells, healthy cells. So it's also my aim to the device can be adopt different cancer cells. And also this device can utilize 100% the drugs. It cannot like wastage of the drug. So first here, I tried to develop a magnetically controlled drug in which we have used iron oxide nanoparticles, and it functionalized with APTES. And after APTES functionalized on the ATPS, we used curcumin as an anticancer drug molecules. And here, this is the FESM and time image of the developed this drug molecule. And actually, further, we have also incorporated one carbon dot. Due to the carbon dot, also it contains some fluorescence property, so that we can also find out how it is traveling out. So here, this is the different characteristics like XRD and FTIR. But if you look about the VSM characteristic, after loading the drug, the magnetic property are getting down, but still it having some magnetic property. And in XRD here, we can see that there is no any change in the peaks due to that after loading the curcumin. Due to that, we again do the FTIR. In FTIR, we can see that after loading the drug, there is some reduction in the peaks like C double O bond and NH and CO. And here also we can see that how the drug is released with respect to time, which is showing a good results. And we have tested the cell viability against the NIS3 cell. So this is the first device which I have just prepared in order to find out what is the impact of the drug in the flow and in trap. So this device consists of some pattern magnet. The pattern magnet having there is some gap between the two kind of magnets. This magnet is prepared by the neodymium iron boron based micro powders. So here what is happening due to the gap in between the two magnets, there is a consecutive difference in the magnetic field gradient. And as we know that if the change in magnetic field with respect to distance will be high, then definitely a more and more larger magnetic field gradient will be generated. The most important thing is pattern magnet is that in a same channel it can give the attraction and repulsion force. So the drug will be not like just flowing and it will trap. It will move like oscillatory motion because it contains some positive and negative magnetic forces. So this is the fabricated device, and this is the magnetic field versus magnetic field gradient curve. So here we can see that up to the lower flow rate of 0.2 microliter per ml, the drug localization was at a single place. That is the interesting thing we can see that. And as we increase the flow rate, still the localization at a single place, but the width of the lateral width is increased. So according to size of the tissue or size of the some cancer tumor, we can modulate the lateral width of the trapped drug. So here we can see that according to change in the flow rate, how it's changing the distribution of the trapped drug. And also here we can after that we find out what is the effect of that localized drug on the cancer cells. Here according to flow rate, if the drug trapping is only 100 mg per ml, so how the cancer cells are killing, and as we increasing the entrapment of the drug, how the cancer cells profile is killing. So in the improvement of the previous device, here we have fabricated a different device in the addition of that particular device, in which if we inject a single concentration or single dose, then it will differentiate in a various concentrations in a single device. And also a device contains to adapt to different cancer or any kind of normal cells. Here there is outlet wells. Here we can see that at the outlet well we culture any cells which we as per demand, and at the inlet we can inject the drug molecules. And there is a magnet in a stair-step-like configuration has been also attached with this. The gap between the channel and magnet is increasing up to from the 50 micron. So it's the first gap is 150 micron and the last one is 200 micron. So here, this is the microscopic image, how the gap is changing according to distance. And this is the FEM simulation, how the distribution of the injected drug is there. Here we can see that as the magnetic field is changing, the magnetic field gradient is also changing. Therefore, attenuating magnetic force will be generated inside the channel, and that attenuating magnetic force is responsible for generating a concentration gradient inside the channel region. That concentration gradient region is only responsible for generating a different concentration at the different outlet wells. And also the main thing is that in the device, the particle not should be trapped, as in previous device, the particle was trapped, but in this device, particle is not getting trapped. It's just deflected, and due to deflection, a longer region of concentration has been achieved. Here we can see that the optimization at what flow rate, how the concentrations are differentiated. So here we can see that after compartmentalization, fluorescent image already shows that how in or the outlet, how the flow image is changing. And here we can also calibrate these all the concentrations which have taken into the outlet wells, how it is changing, and it's measured by the ICP-MS measurement, and it's widely explored. And according to this experiment, we have also found that if we inject, MCF7 has been cultured onto the outlet well, so how it is describing the effect of that cell lines. And also we validated the device by culturing different cancer cells outside the device. We can see that how similar the results are coming in device and outside the device. So it's a summary. We have developed the anti-cancer drug, and we have also designed and fabricated different devices. The application of the device is for IC50 calculation, drug dose screening, and also targeted drug delivery. And in the next work, what we have doing, we have fabricated some membrane, and also we are going to integrate that membrane so that we can find out what is the mechanical impact onto the cancer cell during the treatment. And also we have fabricated a micro heater and also some interdistal electrode so that we can find out in a single chip how the cells are responding against the different dose of concentration. So these things we are going to do, and these are my lab members. So I thanks to everyone. And